Good afternoon, everyone, and welcome to the Infectious Disease Epidemiology Seminar Series brought to you by the Center of Communicable Disease Dynamics at Harvard. So I'm Siyang. I will be your moderator for this session, and I'm very excited and honored to welcome Professor John Drake, who is a distinguished research professor in the Odum School of Ecology at the University of Georgia. So before we started, just a few standard logistics. So the seminar will be recorded as everyone just here. So please turn off your camera if you do not wish to be recorded. And the recording will be available on YouTube and on our website at a later point in time. And after approximately 45 minutes, we'll hopefully have some time for questions. And please send your questions via the chat function to Bradford throughout the talk. And you have the possibility to ask questions to our presenters directly. So in that case, please also notify Bradford or me in the chat. So we'll ask you to turn on your camera and ask us questions by the end of the talk. So now I will introduce our speakers. So Professor John Drake is the director of Center for Ecology of Infectious Disease at the University of Georgia and distinguished research professor of ecology in the Odum School of Ecology. So he holds a PhD from the University of Notre Dame and has been with the University of Georgia since 2006. And he was the Lavingham visiting professor in the Department of Zoology at Oxford University in 2012. And his research seeks to understand the dynamics of biological population and epidemics, focusing on bringing experimental and observational data together with mathematical theory. So some current projects concerns the dynamics of Ebola viruses in West Africa and the spread of white nose syndromes in bats, proliferation of SARS-CoV-2 viruses in the United States and globally, and the development of theory for future infectious disease early warning system. So the title today will be entitled Early Warning Signal of Emerging Infectious Disease. And thank you so much for joining us. Please take it away.
Yang, thank you for that very kind introduction. Let me share my screen here. And as we begin, I'd like to first acknowledge a wide range of collaborators who have assisted with the projects that we're talking about here. Many of the ideas are theirs, and much of the work was done under a contract with NIGMS through the MIDAS network. And so that was the funding source for much of this work. Really what I want to talk about today is a kind of a blue sky idea that came to us about 10 years ago. This concerns a property of complex systems. Complex systems, of course, are those that have multiple parts, often multiple spatial scales, interconnections, feedbacks, and other nonlinearities. Really just a cartoon to indicate what we're talking about. I love this image by Eric Barlow showing a nested food web in an ecological system. But of course, epidemiologic systems are complex systems too. And what I want to talk about today is tipping points in epidemiologic systems and some theory about how those tipping points might be anticipated by subtle signatures in the data. So in ecology, we often use low-dimensional, nonlinear dynamical systems in order to understand the dynamics of ecosystems. And some time ago, maybe 15 years ago or so, it was rediscovered that such systems, when excited by noise, sometimes exhibit some curious properties. And those properties are generated from the combination of noise and the attracting and repelling states of the system. So some models that exhibit this include a model of lake eutrophication and the famous Rosenzweig-MacArthur model for the cycling of biological populations. We oftentimes represent the properties of such systems conceptually using something called the potential function. And the potential function is going to be important to what I want to share with you today. The potential function, sometimes also called the ball and cups diagram, just describes the tendencies of the system. So on the left-hand side here, we've got a model for lake eutrophication. This is what we call a double well potential. There's two quasi-stationary states, a model with low levels of eutrophication. Let's go back. A state with low levels of eutrophication and another state with high levels of eutrophication. Of course, these are separated by a potential barrier that separates the two. And these alternative stable states are interesting, and there's different ways that we can cross from one state to another. Well, one way that we can change from a low to a high state, or vice versa, is if the system gets perturbed sufficiently to move across that potential barrier. But another way is if the system itself is undergoing a slow change, and that slow change results in a morphing of the potential barrier and its disappearance. And this disappearance is what we colloquially refer to as a tipping point in a dynamical system. And so much of what I have to talk about has to do with the creation and disappearance of such tipping points in epidemiologic systems. This understanding of tipping points is associated with a phenomenon known as critical slowing down. Critical slowing down is a very general phenomenon in which a return to steady state after a perturbation becomes slower and slower as the critical point is approached. Ecologists refer to this as a loss of resilience because the system is not robust to various perturbations. And so here you see on the right-hand side this double well potential again, but one that's undergoing a deformation from a system of high resilience, where a perturbation to the ball in that lower equilibrium very quickly returns to its previous steady state, and one of low resilience. And of course, you can see that this is associated with the change in the curvature of that potential function. But the curvature of that potential function is exactly what drives the characteristics of the fluctuations. For instance, the variance is a characteristic of those fluctuations, as is the autocorrelation function. Now, this caused Martin Scheffer and many other scientists around the time to recognize that because of this general property, it might be the case that we could anticipate critical transitions just by measuring statistics like the variance and the autocorrelation function without actually having a detailed knowledge of the underlying dynamical system. So here are some data from an infectious disease in California. This is whooping cough. And whooping cough is a vaccine preventable illness. And due to increases in the number of people refusing to be vaccinated, whooping cough, like other childhood infections, has witnessed something of a resurgence in the United States. And so what you see on the top panel is some data on whooping cough prevalence in California. For a long period of time, we see something that looks like a quasi-stationary state with very little transmission, primarily imported cases and maybe small clusters. But then in 2006, there was a very large outbreak. And the question we have is, could this large outbreak, this movement toward a supercritical population, have been anticipated just by observing the characteristics of this long series of cases leading up to that point? As a motivating example, I'd like to introduce what I call the hinge model. The hinge model is an SIR epidemic, so a low-dimensional dynamical system, probably familiar to most of you, in which at some point, so it begins in a subcritical state, and at some point, we allow there to be a slow drift toward the critical point at R0 equal to 1. This slow drift might be due to an increase in the number of susceptibles in the population, such as through vaccine refusal. It could also be due to other changes, like slow change in the transmissibility of the pathogen, something like that. And what you see here is that, very similar to the example of whooping cough in California, In the subcritical state, there are sporadic cases when there's introduction of the agent into the population. There's a slow drift up toward the critical point. Even during this slow drift, it's not obvious that there's actually much of a statistical difference in the sporadic cases. Once the system becomes supercritical, it's prone to outbreaks. There's a delay, which is referred to as bifurcation delay, and then there's an outbreak of some seriousness. And our question was whether or not this property of critical slowing down occurs in disease dynamics, and could it be used to provide model-independent indicators that this critical transition is being approached. We have studied this phenomenon now in models and in data for about the last 10 years. And what I want to report on today is some of our discoveries. It's turned out that, in fact, there is this phenomenon of critical slowing down in epidemics, but it's actually much richer and, in my opinion, more interesting than I'd ever imagined when we started out on this journey. So this is the SIR model that will be motivating much of what I discussed today. This is the compartmental version of it. Very often, you see these equations represented as differential equations. We make a couple of modifications to the usual model. One is that we allow there typically to be infection that happens spontaneously, and that's indicated here by eta S. And so there's a force of infection from outside the local population, which is basically just reintroduction of the pathogen in a subcritical population. And we allow some of the parameters, such as the vaccination rate, to be time dependent. This is what I want to do is build up some intuition about this system. And so this is the starting point for thinking about that. So what we have here is a sketch of what the potential function for the SIR epidemic looks like. On the lower plane here, what you see is the bifurcation diagram. And so when we're talking about dynamical systems that have equilibrium properties that differ according to what the parameters are, we indicate what those equilibria are, the stable equilibria, by a solid line, and the dashed equilibrium by an unstable equilibrium by the dashed line. And so you see over here we have for some systems where the transmission rate beta is large enough, we have an endemic equilibrium or an interior stable equilibrium. But of course, when transmission is too small, then the disease-free equilibrium is in fact the stable equilibrium. That would be for a deterministic system. For a stochastic system, one which is excited by intrinsic or extrinsic noise, there will be fluctuations around this stable tendency. And those fluctuations depend a great deal on the shape of the potential function. And so the potential function will be steep up here, where the transmission rate is quite high and the endemic equilibrium is large relative to extinction. And the fluctuations will be quite small here, where the disease-free equilibrium is quite stable. But somewhere in between, where either the disease-free equilibrium or the endemic equilibrium is attracting, the fluctuations will tend to be larger, and that's a result of critical slowing down. And from our linear stability analysis, we understand that the characteristic return time diverges as we approach that tipping point where the basic reproduction number equals 1. That's kind of like the core idea behind these early warning signals. But as we've discovered, the situation is much more complicated than that. So here's another picture of that bifurcation diagram. In this case, it's drawn with an eye toward understanding the effect of the immunization level that's required to eliminate pathogen in a population. So again, you see here in a plane showing the immunization level and the prevalence, the disease-free equilibrium here at sufficiently large immunization level, and the endemic equilibrium here at where immunization is below the critical threshold. What these points illustrate is the state of a system after perturbation as it's returning to that equilibrium over time. And so each black dot represents a time point taken 1 year from each other. And the gray dots represent weeks. And so, for instance, when immunization is very high in a population, if you get a cluster, you expect it to die out quite quickly. And so there's a large gap between these points as they return quickly to that disease-free equilibrium. So also then we can see the speed at which different populations might approach the equilibrium, depending on the parameter, the immunization level parameter. It's also the case that this gives rise to underdamped versus overdamped dynamics, where underdamped dynamics are those which approach the critical point through a spiral. Here's another version of that picture. You can see again the stable equilibrium indicated by the solid blue line. And if we perturb the system from the blue line at some point, such as this black point, we can see for different parameterizations of the system, its approach. So it can cycle into the endemic equilibrium here at a speed, again, indicated by the spacing among these dots. Of course, under other conditions, for instance, where immunization is sufficiently high, that system will actually approach the disease-free equilibrium in a trajectory that, again, depends, whose speed depends on the characteristics of transmission and immunization. This particular model is parameterized for a measles-like epidemic. So that's elimination. Here is a cartoon version of disease emergence. So in this case, we have disease emergence not due to vaccine hesitancy in a population which had previously been eradicated, but we have the approach to critical slowing down based on immigration in the population. Immigration is not constant, but fluctuates according to demographic stochasticity. And again, in this example, we have a period of critical slowing down between when immigration begins and when we have that first post-critical outbreak after a period of time due to the bifurcation delay in the system. So those are kind of our basic concepts that we want to try and illuminate. And in the course of a number of investigations, we learned a great deal more about how this critical slowing down happens and whether it's observable or not. Here in this case, what you see is the critical immunization level, the cycling around the attracting point as the critical immunization level is approached. Our first task was to develop some theory for this. This was done in collaboration with Suzanne O'Regan. We used the Van Kampen expansion of the SIR epidemic around its quasi-stationary state in order to derive formulas for what the power spectrum and the autocorrelation, the variance and the coefficient of variation for a variety of simple dynamical systems. This illustrates our general approach, which was to develop some mathematical theory in a limiting example, to then numerically study that theory, and then ultimately to try and demonstrate that data that we have are consistent with that theory. What these figures show here is that comparison of the theory and some numerical simulations. The numerical simulations are shown in black and in blue, and different versions of the mathematical theory are shown in green and in red. One of the early discoveries is that oftentimes around the critical point, the mathematical theory and the numerical analysis do not agree with each other. And so that is an important concern as we think about operationalizing this theory in the service of early warning signals. Here we show both the elimination and the emergence case, where elimination is considered to be achieved through vaccination uptake, and emergence due to increased recruitment in the population or increased transmission. And you see that these different ways to approach the critical point actually give rise to different theoretical values for the coefficient of variation, the variance, and the first order autocorrelation. We wanted to understand whether or not these could actually be used then in an anticipatory way, which is to say, without making use of the model equations, could we just measure these statistics, the coefficient of variation, the variance, and the autocorrelation coefficient? These are customarily done with a moving window analysis in which, as you approach, as time goes on, you measure the sample statistics within a time frame. And as that time frame gets closer and closer to the critical point, one expects to see drift in the parameters. We use the receiver operator curve, and in particular the AUC, which is the integrated area under the receiver operator curve, a traditional statistic used for characterizing the performance of a classification system, to ask whether or not the drift in these parameters actually is sufficient in order to predict the tipping point during elimination or emergence. And what we found in this first study is that, in fact, elimination was readily predicted by CV, variance, and autocorrelation, but it was much more difficult to anticipate emergence, something that led us over the subsequent years to seek to develop additional statistics that are based on these this same phenomenon of critical slowing down. So that's what I think of as the general theory of critical slowing down for epidemics, but there are a number of problems with it. First of all, is the theory is a limiting theory, and so it only works if there's a very slow drift toward the tipping point. In fact, we often have what's much more commensurable timescales between that slow drift and the timescale of the epidemic itself. So that's the fact that the changing average state is really too fast for our quasi-stationary assumptions. The second is that the original theory is based on a continuum approximation, that Van Kampen expansion that I mentioned, which is really just a technique from statistical physics. But biological systems behave according to a discrete state space and the intrinsic noise generated by demographic stochasticity. We needed to develop theory for that. The original theory is based on observations of the system at its true state, which is to say the number of infected individuals in the population. But in fact, our data reporting systems don't have that. Our data reporting systems are case reports that are typically aggregated, perhaps daily, weekly, or even monthly, and are subject to reporting error. Finally, there's both the challenge and the promise of spatial extension and spatial heterogeneity. The challenge is that movement of individuals in space could corrupt the signals of local systems for which we can derive the appropriate theory. But the opportunity is that we have a lot more data signals to study, and maybe the additional degrees of freedom by observing an epidemic in space might provide some increased sensitivity to anticipating critical transitions. And then finally, the issue of seasonality, which one could think of as fluctuations that may go across the critical boundary at R0 = 1 on an annual timescale. And this would be true, for instance, for vector-borne diseases, but also for directly transmitted infections such as SARS-CoV-2 or influenza. So our general approach then is to try and develop more mathematical theory, then simulate more realistic models where the limiting mathematical theory can't be assumed to apply, to compare these to understand where the mathematical theory might be misleading, and then to hunt for critical slowing down, first in simulated data, to look at the prospects for detecting it in the real world. And I'll close with a couple of examples where we find critical slowing down in real data as well. So our first challenge was to understand the effect of a discrete state system. We did this by deriving the relevant statistics here, the mean, the variance, coefficient of variation, correlation time, and autocorrelation for an approximation, which is the birth-death immigration process. This is a good model to approximate emergence under conditions where susceptible depletion is not sufficient to result in a significant change in the force of infection. We also derived a new statistic at this time, the index of dispersion. And for illustration, we compared the estimate of the basic reproduction number that we get under the birth-death immigration process, the approximation, with the true value, again under an emergence simulation, which is modeled on measles outbreak. And we find that, in fact, the maximum likelihood estimate obtained from the birth-death immigration process, the approximate model and the true model agree to a very high degree, which also then suggests for us yet another potential early warning statistic, which is the likelihood ratio between a model for birth-death immigration process with a slow drift, which is to say increasing birth in the population, versus one that is stationary. In fact, that statistic we found to be quite an effective indicator of the approach to the critical transition. I recognize that this is a bit of a complicated slide, but understanding just a piece of it is helpful. So here on the left-hand side, I have to remember that I can't use the mouse. Here on the left-hand side, I have the mathematical theory, which is indicated in red, and the numerical solutions, which are shown in green for three different scenarios, depending on the way that we do the analysis. And the vertical columns show the speed of the approach to the critical point. And the key point is that the autocorrelation, the index of dispersion, and the variance, when the approach to the critical point is fast, are all lagging, but in fact behave according to as expected by the model. On the right-hand side, you see examples of where we actually simulated the system. On the top row, you see the approach to the outbreak, whether that approach is fast or whether it's slow. And on the right-hand column, the stationary case where there's no approach to the critical transition. And in the various rows below that, you see the new early warning statistic based on that difference in log likelihood, an estimate based on the basic reproduction number, and the change between the basic reproduction number and the effective reproduction number, all as early warning statistics. The second challenge was to address this issue of imperfect data. So again, we simulated epidemics. We corrupted the data in two different ways. And then we sought to measure the detectability of critical slowing down. I want to elaborate a little bit on what these problems of imperfect data are. So the first problem is the problem of data aggregation. What we show here is a simulated epidemic. Each of the horizontal lines is a case, of the left-hand side indicating the time of infection, and the right-hand side indicating the removal from the population. And then those cases might be aggregated at different intervals. So they might be aggregated at weekly reports, which are indicated by the vertical blue dotted lines, or they might be aggregated at monthly reports, which are indicated by the red solid line. And so, of course, the actual number of daily new infections, shown in the top in black, shows much higher frequency fluctuations than weekly reporting or monthly reporting. And so, of course, for an early warning system that's based on the statistical characteristics of those fluctuations, you can see why data aggregation might prove to be a challenge. And then, of course, these aggregated reportings, again shown in blue for weekly reports or red for monthly reports, they might also be subject to misreporting. In this case, we're thinking underreporting, where each case is not reported with independent probability. And so we simulated both the truth, the black line, and what the observations might be, the blue line or the red line, for a range of different conditions. So what this plot shows is 4 different scenarios that differ with respect to what the infectious period is. So the top 2 panels are for a case where the infectious period is of a week, and on the bottom 2 panels where the infectious period is of a month. And so then we have infectious periods that are either longer or shorter than the reporting period. On the left-hand side, we've got those cases that are aggregated weekly, and on the right-hand side, aggregated monthly. And the bars range from a reporting probability that's quite low, at around 1/100th of cases being reported. You might think of something like polio or something like that, and a reporting probability that's quite high, ending at more than 50%. The colors correspond to the area under the receiver operator curve. Again, that statistic which we use to describe the reliability of a classification system. And each bar corresponds to a different statistical early warning indicator suggested by theory. So basically, in this space, the blue colored regions are those that perform well, black are those that perform poorly, and brown are those that are positively misleading or inconsistent with theory. And interestingly to us, mean variance and a new statistic that we described for the first time in this paper, the first differences of the variance, actually performed quite well unless the reporting error is really, really high. Similarly, autocorrelation, covariance, and the decay time performed well, provided that the aggregation period of the data is larger than the serial interval and that the reporting error is not too highly overdispersed. And then finally, coefficient of variation, skewness, and kurtosis were found to be unreliable indicators of emergence. And actually, that's a general pattern that we found, and one that's really more of a technical point, but I could discuss later if that's of interest. So the key finding then was that overall, 7 out of the 10 proposed early warning statistics actually performed quite well for very realistic reporting scenarios, which was the first time that we thought that in fact imperfect epidemiological data may not be a barrier to using early warning signals for a variety of potentially emerging diseases. This slide illustrates one of our studies from a spatial model. We were first clued into the possibility that spatial correlation itself might be a leading indicator of catastrophic shifts from some research on spatial transitions in savanna ecosystems and in other ecological systems. One of the things that we found is that the largest eigenvalue of the covariance matrix of the observed state. So now we're not looking at the covariance over time, but we're looking at the covariance over space, becomes much larger when a system approaches a critical point, and the spatial system itself will convert, will flip all at once. We've studied this for an ecological model, which is actually the focus of our paper because it contains some more interesting dynamics, but also for the SI epidemic that's distributed in space. We've subsequently developed this in some incomplete work to ask about metapopulations of epidemics in which there is connection between different populations and immunization, either through vaccination or natural immunization. And one of our early findings there is that the correlation function among different locations might actually turn out to be an important early warning indicator for spatially distributed infectious diseases. And then the last of these challenges that we sought to understand has to do with seasonal forcing. So again, seasonal forcing was considered to be problematic because the period of seasonality is commensurate with the period of the slow drift toward the critical point. We show 3 examples here. On the top, what you see is the effective reproduction number as it changes over time due to its non-seasonal component, and then where you have low seasonality, so small fluctuations on an annual basis, and high seasonality. And of course, the question is, can you actually understand from the fluctuations in the number of cases over time when the tipping point in that non-seasonal component actually occurs? And so in the central set of plots, what you see is a simulation of incidence. And on the bottom, the wavelet analysis showing at different frequencies the power associated with this, this seasonally forced system. This caused us to look at 2 new statistics, which we defined. One we called wavelet filtered reddening, which is just the variance of the time series that's filtered for those selected frequencies, such as the high frequency fluctuations, which might show the approach to the critical point, as well as wavelet spectral reddening, which is the median scale of the spectral density at a given time, which basically quantifies the dominance of the low frequency components of the power spectrum. Those low frequency components are those that correspond to this, the critical slowing down, the fact that as the critical point is approached, the resilience of the system is lost. Again, a heat map to show under some different kinds of approaches to data analysis, the effectiveness of these different statistics at anticipating that critical point. Once again, we find that across all of these different approaches to detection in data, wavelet filtering, the variance convexity, and the index of conversion all actually show very high detectability. You're starting to detect a trend here. Skewness, kurtosis, and the coefficient of variation do not anticipate the approach to the critical point in the periodically forced epidemic. Interestingly, our new statistic of wavelet reddening didn't actually perform very well, while wavelet filtering did. Something that we would like to understand further in the future. All right, that's the theory. What I want to do with the last few minutes is just describe for you a couple of our attempts to measure critical slowing down in data. I was honored when our first work on the theory came out that newspaper reporter asked Steve Strogatz what he thought of the idea. Steve Strogatz is one of the greatest living applied mathematicians, in my opinion. And that he would take the time to comment on it, I found quite flattering. On the other hand, his comment was, I don't think we're ever going to see it, because the messiness of the real world might interfere so greatly with the fluctuations due to critical slowing down. The first system in which we look for critical slowing down has to do with the resurgence of malaria in tea plantations in East African highlands of Kenya. So this is a population which has historically received a high level of medical care, and malaria was effectively eliminated. It's in a critical zone where the climate is really not quite suitable to malaria due to its high elevation. But there was a resurgence in the 1980s and 90s of malaria in this population. The causes of that resurgence are somewhat debated. At one point, it was attributed to climate warming, but it also appears that evolution of chloroquine resistance played a role. And so a student of mine, Mallory Harris, was interested in this system, not so much in order to understand the causes of the resurgence, but to ask the question, could the resurgence have been anticipated just from understanding the fluctuations in the number of case reports over time? And so what Mallory did is she computed a variety of these candidate early warning signals for this system over the period that chloroquine resistance and climate warming were occurring, but prior to the first large outbreaks. And what I'm going to show on the next slide is the p-values for the correlation coefficient between those statistics and time during the course of this approach to the critical point. And so the black lines indicate for each of these statistics that p-value. And so as it approaches the critical point, what we would expect to see is that p-value to go down. The dotted dashed line is a p-value of 0.01 in order to account for multiple comparisons. And the red dashed line, the heavier dashed line, is the standard alpha of 0.05. What I want to draw out is that variance in the upper left, the covariance, and the autocorrelation and decay time all exhibit this property of declining right before the critical transition is approached. The index of dispersion was all a marginal predictor. And these other statistics, which we've discussed before, the coefficient of variation, skewness, and kurtosis, are in fact poor indicators that a critical transition is about to occur. The second example is from the resurgence of monkeypox in the DRC in the late 1980s. So monkeypox is cross-reactive with smallpox. The global smallpox elimination campaign eliminated smallpox at a relatively late time in Central Africa. As a result, the reported cases of monkeypox also went down. But due to the cessation of smallpox vaccination, we can anticipate then that the monkeypox system would return to a critical point at some point. Now, monkeypox is maintained in a natural animal reservoir and typically considered to be subcritical for human population. But the combination of spillover from the animal reservoir and clusters of monkeypox cases with onward transmission result in there being a continuous sequence of cases. The outbreak investigation began in the 1970s, but was followed by intensified surveillance in the 1980s due to the importance of verifying that any poxvirus cases were in fact not due to smallpox, but due to monkeypox, which led to the large number of cases being reported here. So on the x-axis, we again have the number of cases, in this case reported monthly over time. On the y-axis, we have the number of cases, and the line shows the estimate of the variance in a moving window as we approach the bifurcation where large clusters became common. At this time, we were also looking at a new statistic which had to do with the convexity in that the shape of that variance function. According to the linear stability analysis, we expect that the variance would diverge hyperbolically as the critical point is approached. You can kind of see that actually in this reverse L shape for the estimated sample variance. And so we added to this a test for whether or not that variance changed in an accelerating way over time. And places where the variance was found both to increase and increase in this convex way are indicated in red. So what you see is that in the late 1970s, where there is insufficient susceptible replenishment to move towards a supercritical point, there is no such convexity. But the variance does increase during the late 1980s and approaching the time that we would say that the tipping point was crossed. And the last example is hot off the press. It was published yesterday in the Proceedings of the Royal Society. This had to do with an attempt by a number of different groups to anticipate the resurgence of COVID-19 using the same kind of statistics. Results that are published now are a mixed bag. Some people say that they've detected it. Other people say they haven't. We're on the we haven't side. I was approached by Fabian Doblander last year in order to assist with this study. And when he came to me, he said, well, I think we're going to see critical slowing down in some countries, but not others, because they seem to approach this quasi-stationary state in the summer where clusters are just due to imports. And I thought, I don't think we're going to see it because I think we're still looking at the tail of the springtime epidemic. And that tail is going to have, is basically a perturbation an order of magnitude larger in size than what we would see due to critical slowing down. And so we adopted our usual approach. We developed a conventional approach to defining when the lull in number of cases was. That started at some time and terminated with the resurgence. And then in a moving window, we calculated about 10 different early warning statistics. And we looked at whether or not those statistics changed as the critical point was approached. On the right-hand side, what you see is the correlation coefficient for all of the different countries we looked at, for all of the different statistics. Those correlation coefficients that are in red are those that are statistically significant, and those that are in black are not. But the key finding is that almost all of these statistics are on the left-hand side of the number line, less than 0, meaning that the variance, the coefficient of variation, the dispersion coefficient, the autocovariance are in fact declining over time, exactly the opposite of what the theory of critical slowing down says they should, and consistent with the idea that a long-lived transient, i.e. the tail of the epidemic, is actually what's driving the fluctuations. We subsequently conducted a simulation study in order to demonstrate that this is in fact what explains this pattern. I remain skeptical of studies that claim to show, to anticipate critical slowing down in COVID-19 waves. Just to close, I want to mention to you a couple of serendipitous discoveries that came along the way. All of these discoveries also have to do with the behavior of epidemics near their critical points. These have to do with transience, an interesting quantity called the second factorial moment, and also something that we learned about equation-free systems that's also relevant to anticipating tipping points in epidemics. So transience is an interesting phenomena that's gotten some attention by folks interested in dynamical systems, particularly in ecology, and for multidimensional systems that are non-normal, which is to say where the eigenvectors are not orthogonal around a critical point. It's possible that the approach to the critical point may actually deviate from the current state of the system prior to approaching that critical point. And this phenomenon is called transience. Reactivity is a measurement of the extent of transience, or how large that transient might be. So actually, transience is easy to understand in epidemic systems. An outbreak is a transient phenomenon as a supercritical system goes from its disease-free equilibrium at the time that there's an introduced case through an outbreak and back to the disease-free equilibrium as the susceptible depletion causes the epidemic to curtail. We used this measurement of transience or reactivity to look at whether or not the reactivity in the SIR system, its tendency to give rise to periodic fluctuations, actually itself can be used as an early warning indicator. And both for emergence and elimination, we found that transient indicators of tipping points are actually nearly as reliable as the other indicators we've been looking at classically, the variance, the covariance, the lag1 autocorrelation, and so forth. The second serendipitous discovery has to do with this statistic called the second factorial moment. And the formula for the second factorial moment is just reported here. It's just a sample statistic. It's something that we can quantify from a sequence of data. The reason it's of interest to us is because the second factorial moment allows us to distinguish between what looks like an increase in the number of cases due to an increase in reporting intensity versus due to an increase in the transmission rate. So in the middle panel, you see two simulations on the top. The simulations, to my eye, are indistinguishable from one another. On the left-hand side, we have a system which is entirely stationary, but shows an increased number of cases because reporting increased. On the right-hand side, we have a case which is not stationary, and reporting didn't change, but the number of cases actually did. And of course, in both cases, the mean number of cases is going up over time. What's important is that the second factorial moment doesn't change in the first case, but it does in the second case. And so again, we performed a number of simulation studies to see how sensitive this was to various assumptions about the, for instance, the transmission rate and the approach to estimating this sample statistic. This is a theoretical finding that, as far as I know, has never found any application in practice, but I think is really promising. And as we build increasing real-world reporting systems, perhaps important to include in our digital data systems. And then the last has to do with a nagging concern we had, which is that the phenomenon of critical slowing down, we understand very well in low-dimensional systems. We can derive mathematical formulas about it. We can simulate it. We can understand the relationship between the numerical analysis and the mathematical solutions. But there's really no such thing as critical slowing down outside of those low-dimensional nonlinear dynamical systems. But the world's really complicated. And we wondered whether or not more complicated models, what we called high-dimensional models, might actually not exhibit critical slowing down because of all of the different perturbations in many dimensions that are induced by demographic stochasticity and environmental noise. We are not agent-based modelers, and so we teamed up with a couple of different groups, including the group that developed the Fred model, as well as Alex Vespignani's group, to look at whether or not critical slowing down like phenomena might occur in large-scale agent-based models, just as they do in low-dimensional nonlinear dynamical models. And so we actually studied 5 different models, 6 if you count the birth-death approximation that I mentioned earlier, which are all different. They range in complexity from very simple to very complex. And we sought to parameterize these models so that they all were behaved like a system where you had measles that had been eliminated, an increased number of individuals susceptible in the population due to vaccine refusal, and resurgence upon reintroduction of the pathogen. And what you can see here in the bottom left is for all 6 models, the autocorrelation function increases, in fact, approaches 1 as that tipping point happens. Tipping point is defined differently for a system of equations versus an agent-based simulation, but the behaviors are the same, which was encouraging to us that all of our dynamical systems theory would provide useful insight into what is happening in the real world. So in summary, this theory that we've been working on predicts that transmission of tipping points should exhibit critical slowing down. In fact, we think they should exhibit critical slowing down in all kinds of systems, whether they're spatial or seasonally forced, or even these high-dimensional systems, which may behave more like the real world. The devil is in the details, and the details are how you try and estimate it. Critical slowing down, we think, could be used as a model-independent early warning signal. We're optimistic that at some day in the future, there will be early warning systems for disease emergence. But we don't believe the science is there yet, and we're eager to continue pursuing this line of inquiry. But we still believe that it has promise. And this is largely due to our simulation studies, which we have attempted to corrupt and perturb and modify in as many realistic ways as possible. And yet we still find that we detect critical slowing down when we know how to look for it. And then finally, these last examples that I showed you, malaria in the East African highlands and monkeypox in the DRC, we believe are the first examples documenting critical slowing down in real world data streams. We're eager to pursue this further and would welcome any suggestions you might have of appropriate systems in which to investigate the phenomena. So with that, I want to thank you for your time and attention today, for the invitation to speak, and I look forward to engaging in some questions.
Yeah, thank you so much for this terrific talk, Professor Schick. I think in the interest of the time, we'll maybe take 1 or 2 questions. We'll try our best to accommodate as many as possible, but if your question doesn't get covered, please feel free to reach out to our speaker directly. So I guess the first question is from Michael, who want to ask themselves. So please unmute yourself and ask question.
Yeah, thank you very much for a wonderful lecture. My question is about your recent article, which you showed about COVID-19. Have you had a chance to apply this analysis either at a smaller spatial scale, for example, doing this analysis for the states in the US, or alternatively looking at reported hospitalization or death, which presumably are more accurate than the reported cases?
So we have not done it for different geographic areas. I think that's something that could be done. And with respect to other data series, we did spend some time looking into that, but we found that the case reports was probably the clearest and the simplest example. We didn't pursue that analysis in depth.
Thank you.
Yeah, maybe one more question from Bradford.
Sure, thanks. Great talk. I guess I'm very interested with this eta parameter, the sort of the sparking rate of the epidemic again. And I'm wondering about particularly with SARS-CoV-2 transitioning from a more epidemic to endemic phase, whether, and this eta parameter could potentially vary over time, whether or not you've thought about what sort of statistical signals or what you expect from that variation over time.
So that's a good question. I do think that the eta would vary over time as a result of embeddedness in a larger system and how much transmission is going on in other locations. And of course, it might also be related to the coupling between two locations, right? How that affects early warning statistics is not something that we've investigated. I think my approach to it would probably be rather a model-based approach than a model-independent one, and attempt to estimate that parameter as it's changing over time. We have an analysis for SARS-CoV-2 in California, which is available on medRxiv, in which we used a compartmental model that was semi-parametric, and that allowed for the transmission rate to fluctuate over time. I think the same modeling framework could be used to allow the immigration rate to change over time. But we also have to recall that immigration events are unique. And so when they're dispersed sufficiently far in time, I don't think you're actually going to be able to estimate it in that way, right? If the time scale over which there's fluctuations in the theoretical immigration rate is faster than the time scale that immigrations actually occur, I think you won't be able to estimate it that way.
Right, thank you. In the interest of time, I think we can move the discussions offline. So lastly, on the behalf of CCDD and the Harvard Th Chan School of Public Health, let's thank Professor Drake again for joining us today and for this fantastic talk and discussion. Please do get in touch with any of the seminar organizers if you have further questions for us or for the speaker. And also please check out the CCDD webpage to set up dates for upcoming seminars. So next week's seminar will be given by Professor David Potel from the Yale School of Public Health, and he will tell us something about policy modeling for SARS-CoV-2 screening, prevention, and vaccination. So thanks again, everyone, for joining, and hope everyone have a good day. And thanks, Professor Drake, for coming.
Thank you, Siang.